Okay.
Start.
Good morning, students. Welcome back to the another session of plant physiology. This is the last session of chapter 4, photosynthesis in higher plants. And in this session, previous classes, you have learned about site of photosynthesis, reaction center, that is chlorophyll A and accessory pigments, photosystem 1, which is located at 700 nanometer wavelength, photosystem 2, located at 680 nanometer wavelength, and the cycles, photophosphorylation cycles, non-cyclic and cyclic. Both you have learnt. How the light is harvested and ATP and NADPH are synthesized in the chemiosmotic hypothesis. You have learnt in detail. And further, the end products of light reaction, oxygen is liberated, whereas ATP and NADPH are utilized in dark reaction or biosynthetic phase, which was discovered by Melvin Calvin, and the cycle is named after him as Calvin cycle. So in this manner, you proceeded to the further next stage, photorespiration. Photorespiration, where you are seeing when oxygen and temperature is high. Oxygen concentration is increasing and temperature is high. At that time, you're seeing that photorespiration starts. And in this photorespiration, the yield of photosynthesis will be decreasing. To overcome this problem, some plants developed a cycle called C4 cycle, where they'll be having oxaloacetic acid, which is the first stable compound formed. And also crassulacean acid metabolism is also noticed in certain xerophytic plants. These plants also will undertake C4 cycle and C3 cycle together in the mesophyll cells. And you already learned about the C3 plants and C4 plants differences. So overall, the concept of photosynthesis in higher plants is learned by you, but there is still some more part. That is the last part, the factors affecting the rate of photosynthesis. How several factors are helping in the rate of photosynthesis and how these factors are going to have an effect on the rate of photosynthesis. Let us understand this concept today. Before that, I would like to appreciate D. Sunita Y. Anusha from Balanagar TSRJC, B. Ashrita Harika, G. Soumya, Manusha, N. Divya, Abhinayasri, and one more name, Sweta also, she submitted from Vangara TSRJC. Y. Nagasri, R. Triveni, N. Satya Krishna, Karishma, Sandhya, S. Akhila from Vaira TSRJC. Very good students, well done. Girl power. All the girls back to back, they were sending me as early as possible, and I'm very happy with their work, with their dedication. They can be successful in near future. So what I want from all other students is dedication is very important. Simply just learning, anyone can learn. With full dedication, if you learn, you can excel in your field. So BPC is not a small stream, it is a good stream, and there is a lot of scope for your survival with this BPC. So I want you to be studious and very energetic. Next classes will be conducted by Kumar sir and followed by Srikanth sir. So be active, at least now onwards. Be in time, complete the assignments as early as possible and forward it to your concerned teachers so that they'll appreciate, and that appreciation is going to be a motivational drive in you that is going to lead to success. So let us begin with the factors affecting rate of photosynthesis. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. As it is the plants who are the producers, all these plants are undertaking photosynthesis. The more photosynthesis they take, the more yield will be there. And these crop plants are our food. They are providing food. So definitely the photosynthesis rate must be higher and higher, so that the photosynthate, the product, will be high as per the increasing population. Also, it is very much important. Rate of photosynthesis must be high in crop plants. Rate of photosynthesis is influenced by several factors, both internal and external factors. As you can see in a plant, the internal organization you'll be learning in the morphology, where you'll see the importance of these green leaves, these plants, where the light is harvested, where the food is prepared, the glucose is prepared or synthesized, and that will be stored in different parts like fruit, seed, grains, or rhizoids, whatsoever. The food is stored in the plant body and we are consuming, animals are consuming. Either directly or indirectly, we are dependent on plants only. So here, several internal factors will be there in the plant. The plant factors include number, size, age, and orientation of leaves. So it is the green part of the plant, that is the leaf, which is very important, very crucial. The number of leaves, the size of the leaf, whether it is very large or small size, the age of the leaf also, and orientation of leaves towards the sunlight, whether it is getting the sunlight, carbon dioxide, all these things are very much important. And the mesophyll cells which are present in the leaf, the innermost part of the leaf cells, or mesophyll cells, where chloroplasts are present, and internal carbon dioxide concentration, CO2 availability, or the concentration of carbon dioxide internal in the mesophyll cells, and the amount of chlorophyll pigment which will be trapping the sunlight energy. All these are the factors which are present in the plant, and they are the plant factors which are very, very important, and they are going to decide the rate of photosynthesis. As well as the plant factors are dependent on the genetic predisposition and growth of plant. So even the genetic makeup, the chromosomes, is also very important in the plant, and this is going to decide the growth of the plant, its life cycle, everything. So all these are going to have an effect on the rate of photosynthesis. Factors affecting rate of photosynthesis: external. External means not only the internal plant factors are important, but external factors on which the plant is depending, the abiotic components like the sunlight, the carbon dioxide availability, the water availability, mineral nutrients, all these are also very, very important. Some cardinal points given by Sachs. Sachs is the father of physiology, plant physiology. Here, temperature. Temperature, if we see, it is very important. As the sun rises and the rays are going to fall on the green leaves, they start up the synthesis. They start up the light reaction. That is, the chlorophyll molecules are going to get excited by taking the high intensities of light. Okay, so the light intensity, the light is traveling, and the energy, quantum energy present in the photons of light packets, is going to be trapped by the chlorophyll molecules. These chlorophyll molecules, they are having their light harvesting complex or reaction center. That is very important. And sun rays will be having temperature. They are going to increase the temperature, and the temperature, if it is minimum, the rate of photosynthesis is going to stop. That means during the evening time, rate of photosynthesis definitely will stop as the temperature decreases. And temperature optimum means the rate of photosynthesis is going to become maximum, because the more amount of chlorophyll molecules can trap the solar energy, get excited, and start the light reaction and dark reaction will be proceeding. So here the temperature, if it is optimum, as you can see in C3 plants, 10 to 20 degrees centigrade is the optimum temperature for the photosynthesis, and in C3 plants. In C4 plants, it is 30 degrees centigrade. That means at high temperature also. So here the temperature is very crucial, and it is going to show effect on the rate of photosynthesis. So optimum temperature is very important for any plant to undertake the photosynthesis, and at optimum temperature only the rate of photosynthesis is maximum. And temperature is maximum when the temperature is very higher and higher. the rate of photosynthesis will stop. If it is very high also, it cannot undertake the photosynthesis. It will halt, it will stop. So optimum temperature is must. You can take example of yourself. If it is very hot in summer, you can't stay in the refrigerator yourself. Your internal organization, your body parts will cool down and they will not function. And enzyme activity, all those things at cellular level, all those things will not take place. If it is very cool also, you can't sit on a hot pan, because your organ system, cells, all these are going to get affected with high temperature also. So temperature is very important. It is important that sunlight must be there, but if it is very less minimum, it will stop the photosynthesis. And if it is very high maximum, the enzymes will be denatured. Certain enzymes will be responsible for the electron transport and all those things. They will not work properly. So that's why the temperature is very important, and optimum temperature is important for the rate of photosynthesis to be maximum. And Blackman, we have here Blackman's law of limiting factor. Blackman's law of limiting factor, he says that when a process is conditioned as to its rapidity means very fast, quick, by a number of separate factors. And the separate factors here, we are talking about photosynthesis, the internal and external factors. The rate of the process is limited by the pace of the slowest process. Slowest process. It is the factor which is present in minimum amount. So what he is stating that, for example, if we take chlorophyll and light, which are very essential, light is important, chlorophyll is important, hmm? And for example, carbon dioxide synthesis will take place, that is important, water is also important. These are essential things which are most important for photosynthesis to take place. And we have, for example, 10 units of light, 10 units of chlorophyll, 10 units of carbon dioxide, 10 units of water. So okay, done, photosynthesis is taking place. Okay, photosynthesis is taking place steadily. But if one factor, for example water, if we are taking, and we are making water, it is only 2 units. 2 units of water is available. Light is 10 units, chlorophyll 10 units, carbon dioxide 10 units. But here, what is in lowest or the slowest process is water. Water is getting limited here. And when the water concentration is becoming less, it will decide this limiting factor. This water is going to decide the rate of photosynthesis now. Okay. Previously, when light, chlorophyll, carbon dioxide, water was equal, the process was done, it is steadily going on, photosynthesis. But if any of this factor, here we have taken water is getting reduced, getting limited, then it is going to affect the rate of photosynthesis. So this is what Blackman is stating, that it is the law of limiting factor. So we can say that law of limiting factor given by Blackman is nothing but the factor which is in the least or minimum concentration will decide the rate of photosynthesis. The rate of photosynthesis is decided by the limiting factor only. So here also it is given light intensity, rate of photosynthesis. You can see here light intensity. When light intensity is there, the rate of photosynthesis steadily is increasing. As and when light intensity gets reduced or gets limited, then rate of photosynthesis will not further increase. It will be slowing down. Or finally, it may also stop if light is not available also. Okay. So here coming to the light only, external factor light is very important, and the quality of light. Light is the external factor. Sunlight is the main source for plants to undertake photosynthesis, and it is the wavelength. The quality of light means the wavelength. We have the visible light, which will be having VGR 7 colors, and at this blue and red wavelength only, more photosynthesis can take place because chlorophyll molecules are going to get excited more at this blue wavelength and red wavelength. Intensity. Intensity means if light is more or less. Some of the plants are surviving in shade plants. They are called sciophytes, and some are sun plants. They require more and more sun, so they are heliophytes. So intensity of light is also important. Duration. The light must be made available at least 10 to 12 hours for photosynthesis to be at the maximum. So here the duration of light, the availability of light is also very important. 10 to 12 hours light. Now coming to increase of in light intensity. If the light intensity is increasing, rate of photosynthesis increases. But very high light intensity slows photosynthesis and finally it may stop also. So there is an optimum temperature at which the photosynthesis rate will be maximum. If the intensity of light is increasing also, to some extent it will increase the rate of photosynthesis, but very high light intensity, the same process will be slows down or finally it may stop also. Carbon dioxide, it is also very important. It is present in the plant. Inside the plant mesophyll cells, the availability of carbon dioxide concentration will be there. And slight increase in carbon dioxide concentration increase rate of photosynthesis, which you have learned in 10th standard itself, that carbon dioxide, if it is made available more and more, what will happen? To certain extent, the photosynthesis rate will be increasing. After that, beyond that, the carbon dioxide fertilization, this is called carbon dioxide fertilization effect. But beyond that, if more carbon dioxide concentration is there, it will reduce the photosynthesis rate. That means it is called carbon dioxide toxicity. So more amount of carbon dioxide, if you are filling and filling for the plant, it will not increase the photosynthesis, because you know about Blackman's law of limiting factor. All the factors must be present in sufficient quantity. Oxygen. Yeah, oxygen is the end product of light reaction. This oxygen will be present in the leaves after the light reaction. Then it will diffuse outside. Here, increase in oxygen concentration decreases photosynthesis. If more amount of oxygen is there, you know the nature of Rubisco enzyme. RUBISCO. It is carboxylase as well as oxygenase. So the carboxylase nature is very important for photosynthesis. But if it is becoming oxygenase due to higher concentration of oxygen, we will have the photorespiration taking place. This photorespiration is a wasteful process. If it is taking place, it will affect the photosynthesis. So here the oxygen concentration, if it is increasing, it is going to decrease the photosynthesis. Okay, and this is called Warburg's effect. Temperature, as just now we have discussed about temperature, minimum, maximum, optimum temperature. These three are important cardinal points, and how it is going to affect the rate of photosynthesis. Slight increase in temperature, rate of photosynthesis increases. Very high temperature, further the temperature is becoming higher and higher, rate of photosynthesis decreases and stop. What could be the reason? If the temperature is increasing higher and higher, transpiration will be taking place through the stomata, water loss will be taking place. At high temperature, what will happen? Water loss will be taking place, and you know abscisic acid, it is going to close the stomata to prevent the water loss. And if stomata is getting closed, carbon dioxide entry will be stopped. So this is the reason photosynthesis will stop. As the temperature is increasing higher and higher, rate of photosynthesis will be stopping, decreasing and stopping, because the stomata will get closed. Only the slight increase in temperature, rate of photosynthesis will increase. But very high temperature, rate of photosynthesis decreases. increases, slows down, and stops because at high temperature, the stomata are going to get closed by the abscisic acid, which is a plant hormone or phytohormone that is going to close down the stomata to prevent the loss of water by transpiration. So you can see how these are interlinked, how these temperature, water, stomata closing, opening, all these are interlinked with each other. And all these factors are going to affect the rate of photosynthesis. As you know, water is also very important. Decrease in water, photosynthesis slows and stops sometimes. And you know that if water is also less, stomata will close. Stomata will be getting closed by abscisic acid, phytohormone. You can see stomata open and close. Stomata is nothing but the hole, the entry, the gate. And you can see here bean-shaped god cells are there. These bean-shaped god cells are also having the chloroplast in them. And you can see the subsidiary cells which are surrounding the god cells. Okay. And this stomatal pore is there, and you can see vacuoles are also present in the god cell. When the water is going to get deposited in these god cells, they will be getting opened. And when temperature increases to prevent the loss of water, abscisic acid, what will happen? The water from these god cells will be going to the subsidiary surrounding cells and it will get closed in this manner. The god cells are going to get closed. When they are swollen, it will be having a stomatal pore or the opening for the entry of carbon dioxide gas. But here, when the water is lost from these god cells, water is getting moved to the subsidiary cells, surrounding cells. When more temperature is there, at that time, they will come closer like this and it will be closing the pore. Stomatal pore will get closed. That means it is a stomata or stoma closed. This is stoma open. So you can understand the importance of these tiny pouches, tiny holes, stomata, which is very important, which is also going to have which is also going to be the factor affecting the rate of photosynthesis. So opening and closing of stomata is also very important. Coming to the internal factors. Internal factors means inside the plant. Let us see protoplasmic factor. You know protoplasm. Every cell has a cellular content. The jelly-like material which is present here in the cell is a protoplasm. So scientists, it is one imaginary or hypothetical factor. So far it is not yet discovered. This cytoplasm of plant cell is having certain factor that is not yet discovered. That is also having effect on the rate of photosynthesis. So this cytoplasm or the protoplasm, it is also a factor. Certain factor is hidden which is not yet discovered by scientists. That's why it is called hypothetical or imaginary factor. That means internal to the plant only. Inside the cytoplasm, some factor is there that is also deciding the rate of photosynthesis. Chlorophyll content. Yeah, chlorophyll is also very important. You must know the chlorophyll. It is a pigment molecule. It is present in the chloroplast. And the more number of molecules of this chlorophyll pigment is there, more harvesting of sunlight energy will be taking place and photosynthesis will proceed. So this chlorophyll content is also important. Assimilatory number. So here a new term is there for you, assimilatory number. This assimilatory number is nothing but amount of carbon dioxide fixed per gram of chlorophyll per hectare, per hour. That is not hectare. Okay, per hour. Per hour, how much amount of carbon dioxide is getting fixed? How much amount of carbon dioxide is converted to glucose? And how many grams of chlorophyll molecules are required? That is going to be the assimilatory number. It is the assimilatory number of this chlorophyll. Variegated leaves. It is very important to study about the variegated leaves, this assimilatory number, because variegated leaf is nothing but, you can see here, variegated leaves will be having other colors also. Other than chloroplast, other pigments are going to be present in more quantity, and they'll give these patterns of colors on the leaf. So they are called variegated leaf. Money plant is also example. And many of the plants you'll see, show plants will be having these variegated leaves. And here, the more number of chloroplast or chlorophyll pigment, the more photosynthesis will take place. If chlorophyll pigment is very less and other pigments are more, then also it will have an effect on the rate of photosynthesis. Number of chloroplast. Number of chloroplast is also very important. Per cell, how many number of chloroplasts are present? Number of stomata. Stomata will be present on the upper surface and lower surface in some leaves, and in some of the leaves only on the lower surface it will be present. So thus, and some of the leaves like gymnos, you will see sunken stomata will be there. So the presence of the stomata, the number of stomata, and the position of stomata will also decide the rate of photosynthesis. So we are not going to keep the stomata as a place on the plant. It is coming genetically. The arrangement of the stomata, everything genetically it is happening. So because of that only, the sunken stomata or the upper layer stomata or the lower layer stomata, whatsoever the position of the stomata and the number of stomata, these are going to have an effect on the rate of photosynthesis. Now the most important, accumulation of photosynthate. Accumulation of photosynthate decreases rate of photosynthesis. This is very important. What is accumulation of photosynthate? First, you must know what is photosynthate. Photosynthate is nothing but the glucose which is prepared. The glucose which is prepared by the plant or the leaf, particularly in the leaf part of the plant body only the glucose is synthesized. Now, this is going to get accumulated more and more. So it must be moved from there. If it is still there only, then it is going to affect the rate of photosynthesis. It will not take place. It will decrease the rate of photosynthesis. So this glucose, it must be moved to the different parts of the plant body. For example, some stem it must move, or root or a fruit, wherever it has to be stored into the storage tissue, it has to be sent. But when we send this one, this is called translocation of photosynthate. Translocation means the movement of this glucose molecule from leaf to the storage tissues. Okay, that could be anything. The storage tissue can be a fruit, or in the roots, or in the stems, or different parts of the plant body. The photosynthate will be moving, and this translocation has to be done very immediately. So this glucose is very reactive. It is a reactive one, and when it is translocated, it may interact, it may get reduced, and it may interact with the cells of the phloem tissue. So it should not be transported in the glucose form. So it will be converted to sucrose, which is a disaccharide. It is a disaccharide. So here the photosynthate, that is the glucose synthesized in the leaf, will be transported as sucrose, and it is a disaccharide, and it is non-reducing sugar. Non-reducing sugar means it will not it won't react with the cells from where it is getting transported. So here in this manner it will be moving. After that also it will be stored. This sucrose is going to get stored as starch. It will be converted to starch. It will be converted to starch, and it will be stored as starch. Starch is nothing but it is a polysaccharide. Starch is polysaccharide. Polysaccharide means the glucose molecules which are present, no? These glucose molecules, glucose, side by side, they'll be joining by glycosidic bonds. The bond which is going to join them is glycosidic bonds. These glycosidic bonds are going to join by removal of water, by eliminating the water present in the glucose. These glucose molecules are going to get joined by side, and it is going to make the polysaccharide called starch. So this is very important. The accumulation of photosynthate decreases the rate of photosynthesis. That's why immediately the translocation of the photosynthesis has to take place. So assignment 9, tabulate 8 differences between C3 and C4 plant cell cycles. Yesterday we have completed the C3 and C4 plant cell cycles and described the C4 pathway also. These are your assignments. You have to complete your assignments and send it to Kumar sir, P. Kumar sir. Already he has taken enzymes class for you. So if you forward your assignment work to him, then he will be appreciating you in his next session. That is going to be on Thursday. Next week, Thursday. Again, of this, what you have learned. Today's class, you have learned about the fact photosynthesis, and in this you have learned about number, size, mesophyll cells and chloroplast, internal carbon dioxide concentration, and the amount of chlorophyll which is present in the plant. All these are internal factors. Apart from that, even the genetic predisposition, the factor for growth of plant also. So these factors are going to affect the rate of photosynthesis internally to the plants. External factors are also there like sunlight, temperature, and carbon dioxide, water. So cardinal points of carbon dioxide temperature given by Sachs, you can see temperature must be optimum. If temperature is optimum, rate of photosynthesis is maximum. Otherwise, if it is minimum or maximum temperature, rate of photosynthesis will stop, because enzyme activity is also something which is present in the photosynthesis taking place, and sunlight is very important. Blackman, what he states that the limited factor means the factor which is in least or minimum concentration will decide the rate of photosynthesis. Okay, if anything is going to become limited, very less, insufficient, then it will definitely affect the rate of photosynthesis. So this is what given by Blackman's law of limiting factor. External factor, light is very important, and you know the quality red and blue light is very important, and some plants will grow in shade and some in sun. But overall, the duration, 10 to 12 hours duration of sunlight must be available for the growth. And increase in light intensity increases rate of photosynthesis. But very high light intensity, it will slow down the photosynthesis. Carbon dioxide is also very important. If slightly carbon dioxide concentration increases, it will increase the rate of photosynthesis, which is called as carbon dioxide fertilization effect. And more carbon dioxide, it is going to be the carbon dioxide toxicity that will reduce the photosynthesis. For oxygen is also very important. Oxygen is liberated during photosynthesis. If it is more in concentration in the leaf, then it will make the rubisco undertake photorespiration. And that photorespiration is wasteful process. So here increase in oxygen concentration decreases photosynthesis, which is also called as Warburg's effect. Sorry. So here high concentration of oxygen leads to photorespiration. Temperature, slight increase in temperature, rate of photosynthesis will increase. Very high temperature, rate of photosynthesis decrease and stop. As you know, temperature increases means transpiration will also increase. Water loss by the plant body also increases. And you know, stomata will be closed by abscisic acid control. Abscisic acid will make the stomata close to prevent the transpiration, water loss. And if stomata is closed, definitely photosynthesis will stop. Sorry. So here water, decrease in water, photosynthesis slows and stops, and stomata closes. So stomata is also very important factor which may be present on upper surface or lower surface of the leaf, and also there may be sunken stomata in the scene. Internal factors of the plant, the protoplasmic factor, the hypothetical or imaginary factor not yet discovered. It is left for you to discover, student. So chlorophyll content is also very important, and chlorophyll, the more chlorophyll, the more sunlight will get harvested, and assimilatory number is going to be the amount of carbon dioxide fixed by gram of chlorophyll per hour, not hectare, per hour. So next, variegated leaves, in that we can find out the assimilatory number, and number of chloroplast is also important. Number of stomata and the position of stomata, and last but not the least, the accumulation of photosynthate. Whatever the product of photosynthesis, the glucose is getting accumulated, will also decrease the rate of photosynthesis. So immediately it must be translocated. The photosynthate must be transported, where it must be transported to the storage areas, from leaf to storage areas, and that too the glucose will be converted to sucrose, and sucrose will be converted to starch. And this is the assignment, depending on the last class, tablet a difference between C3 and C4 plants or cycles, and describe the C4 pathway. Thank you for listening. Thank you one and all. I would like to thank our secretary sir, who has given this golden opportunity. He made me come in contact with all the students of other colleges. And thank you, sir. And thank you, our DS sir, also for giving this opportunity. And I also I also thank our Sarla madam, who is the coordinator for botany, who was always there for me to appreciate my work as well as to motivate me to take up the classes very smoothly. Thank you, ma'am, and thank you all children who forwarded their work. I hope you will continue the same. All the best for your IP. Thank you. Sir, class is over. Sir? Sir, class is over.